Labeled immunoassays. So labeled immunoassays are designed for antigens and antibodies that may be small in size or present in very low concentrations. You use a labeled reactant to monitor the amount of specific binding that has taken place. And it's used to measure analytes such as bacterial antigens, hormones, drugs, tumor markers, and specific immunoglobulins. Basically, so far we've learned that an antigen will bind to an antibody. And when an antigen binds to an antibody, they form a complex, that antigen-antibody complex. Now, the binding of the antigen to the antibody is very specific. And there's two different things that we can look at in an immunoassay like this. We can either try to detect the presence of an antigen or a specific type of protein or molecule, or we can try to detect a specific type of antibody. Now, if we try to detect the antigen, that means that we're suspecting that that antigen is inside of the patient sample. If we try to detect a particular antibody, then we're suspecting that the person has been exposed and has developed antibodies against the substance that we're interested in finding out about. So as we go through here, all of these are based on the premise that an antibody is going to find and bind to a specific antigen. And when it does that, the antibody is typically labeled, or there's like a marker that's attached to it, and that marker will allow us to detect the attachment of that antibody. And those markers, or those labels as they call them, they can be enzymes, they can be fluorescent molecules, they can be radio labeled, so radioactive materials. There's different ways that we can detect them. And we're going to go through here and learn about some of those different ways that we can detect the antibody antigen binding. So competitive immunoassays, all the reactants are mixed together simultaneously. Then labeled antigen competes with unlabeled patient antigen for a limited number of antibody binding sites. The amount of bound label is inversely proportional to the concentration of the labeled antigen. So if we look at a picture here, here's one sample on the left, and here's one sample on the right. Now the sample on the left does not have the antigen that we're looking for. So unknown concentration of analyte in the patient sample, red dots, competes with the labeled analyte. So these are labeled, and the red dots are not labeled. Now, if the patient has the red dots, if the patient has the antigen that we're testing for, this labeled antigen competitively binds to the antibody. So in here, all of the labeled antibody is going to be bound, and then it's going to produce a really strong signal. Over in this sample, because the patient sample does contain the antigen that we're looking for, some of the labeled antibody is going to bind, or antigen is going to bind, while some of the patient antigen is going to bind. So that reduces the signal or weakens the signal. So as the signal decreases, the analyte concentration increases. So the more of this we have, the weaker the signal we're going to have. So this is what's meant by it being inversely proportional to the concentration of the labeled antigen. Because they're competing for a limited number of antibody binding sites, if the antigen is found in the patient, it's going to take up some of those sites, and then the labeled antigen isn't going to be able to bind. So that's going to be washed away, and this is what we're going to be left with. Non-competitive immunoassays, the antibody is first passively absorbed into a solid phase. Unknown patient antigen is allowed to react and be captured by the antibody. After washing to remove the unbound antigen, a second antibody with a labeled is added to the reaction. The amount of label measured is directly proportional to the amount of patient antigen. Radioactive substances, enzymes, fluorescent compounds, and chemiluminescent substances have all been used as labels. So here we have sample A. In sample A, it doesn't have the antigen. So we have bound antibody on the bottom of the well. So when it says antibody is first passively absorbed to a solid phase, usually that solid phase is like the bottom of a plastic well. And we can see these well plates of like 96 wells. There's smaller ones too, but we would bind these antibodies or absorb these antibodies to a solid phase. And the antibodies are going to be specific. So we know what these antibodies are going to be able to stick to. Then we add the patient sample. Now, if the patient sample contains that antigen, then the antigen is going to bind to those antibodies. So the sample A on the left does not have the antigen. Sample B on the right does have the antigen. So when we add the patient sample, nothing's going to bind here, but the antigen is going to bind here. Then we add this labeled antibody. And as it said in the slides, this label, and as I mentioned previously, can be radioactive substances, enzymes, fluorescent compounds, and chemiluminescent substances. But if the antigen stuck, what's going to happen is these antibodies that are labeled are then going to stick to that complex. If there's no antigen, there's nothing for those to bind to. So during this washing step, these labeled antibodies would be washed away and there would be nothing left to create a signal. But over here, we have the labeled antibody binding. So then when we put in our detection, so if we add the substrate to this or whatever method we're using to detect this, we would see a positive reaction here. So what's going on over here? This is kind of the reverse in which we passively absorb antigen to the bottom of the well. Then we're going to add antibody to it, and the antibody would stick to that antigen. And then we add a secondary antibody, and that secondary antibody is labeled, just like the secondary antibody in these examples. And with this secondary antibody being labeled, it's then going to bind to that primary antibody and then produce a signal. Now, if the antigen was not added, if this antibody was not found in the patient sample, then we would go through this whole process and this capture antibody or the labeled antibody would have nothing to stick to. Radioimmunoassay. This is the first immunoassay that was developed, and it uses radioactive labels. 125 is the most popular. It emits gamma radiation, which is detected by a gamma counter. It's extremely sensitive and precise, and measures trace amounts of analytes such as hormones, serum proteins, and vitamins that are small in size. The amount of label in bound phase is indirectly proportional to the amount of patient antigen present. The disadvantages, though, are you're working with radioactive substances, having to dispose of low-level radioactive waste, and there's a short shelf life of some of the reagents. Enzyme immunoassays are much safer, abbreviated EIAs, and you will see that abbreviation on tests and on the qualifying exam. It uses enzymes and labels which react with suitable substrates to produce breakdown products that may be chromogenic. Chromogenic means color producing. Fluorogenic means fluorescence producing. Or luminescent, and luminescent would be giving off light. Alkaline phosphatase and horseradish peroxidase have the highest turnover conversion of substrate rates and have high test sensitivity and are easy to detect. There's heterogeneous and homogeneous EIAs. Heterogeneous require a step to physically separate free analyte from bound analyte. In competitive assays, it involves enzyme-labeled antigen competing with unlabeled patient antigen for binding sites on an antibody molecule. It's used for measuring small antigens that are relatively pure, like drugs and hormones, and has high specificity. In non-competitive immunoassays, they offer high sensitivity and specificity, simplicity, and low cost. Enzyme-linked immunosorbent assay, or ELISA, is used to measure antibody production to infectious agents that are difficult to isolate for autoantibody testing. HIV, hepatitis B and C, and it's easily applied to point-of-care and home testing. Antigen is bound to the solid phase. Then the patient's serum is added. We know that there's some antibodies in the patient's serum, but we don't know what antibodies. So we know what the antigen is. So like, let's say we're binding the antigen of HIV to the bottom of the well. And then we want to see if the patient's been exposed. So we're going to put the patient sample in there and see whether or not the patient has HIV antibodies. After a wash step, an enzyme labeled antiglobulin, a secondary antibody, is added. The second antibody reacts with any patient antibody that is bound to the solid phase. If no patient antibody is bound to the solid phase, the second labeled antibody will not be bound. After a second wash step, enzyme substrate is added. The amount of color, fluorescence, or luminescence is measured, and this amount is directly proportional to the amount of antibody in the specimen. So for this ELISA that was just described, on the bottom of the well you'd have antigen, and then you would have the patient sample. If the patient sample contained the antibodies against the antigen that we're testing for, so if we put that like HIV antigen on the bottom, and we want to see if the patient's been exposed to HIV by looking at what antibodies are in their serum, if the HIV antibodies are present, that means that they've been exposed to HIV. So those antibodies would then bind. But then we use a secondary antibody, and that secondary antibody would then bind to the primary antibody. And it binds to any human antibody, and it would only bind if it's all bound together. And then that secondary antibody is linked. That's why we call it enzyme-linked immunosorbent assay. It's linked to the antibody. So enzyme is linked to the antibody. So when we add substrate, we're going to see some type of color change or fluorescence or luminescence that happens, and that can be detected by an analyzer. Sandwich immunoassays, also known as capture assays, are best suited to antigens that have multiple determinants: antibodies, cytokines, proteins, tumor markers, microorganisms, especially viruses, also used in the measurement of immunoglobulins. So excess antibody is attached to the solid phase is allowed to combine with the test sample and capture any antigen present. After an appropriate incubation period, enzyme-labeled antibody is added. The second antibody recognizes a different epitope or binding site than the solid phase antibody and completes the sandwich. So you'd put antibody at the bottom, the antigen would be in the middle, and then you would have a secondary antibody bind to a different portion of that antigen, and it would create a sandwich. So antibodies on the outsides and then the antigen in the center. Either a colored or chemiluminescent reaction product is detected. Enzymatic activity is directly proportional to the amount of antigen in the test sample. In a homogeneous EIA, these antigen-antibody systems do not require a washing or separation step. So previously, we would, and we're going to do this during our lab, but previously what we would do is we would bind the substance to the well, and then we would add, like we would wash away any unbound substance, and then we would add that antibody to it, the primary antibody, and then we would let it bind for a period of time, and we would wash away any unbound antibody. And then we would add a secondary antibody and wash that away. In homogeneous EIAs, we don't have to do those washing steps. Enzyme activity is directly in proportion to the concentration of patient antigen or hapten present in the test solution. When antibody binds to specific determinant sites on the antigen, the active site on the enzyme is blocked, resulting in a measurable loss of activity. Homogeneous are less sensitive than heterogeneous. They're rapid and simple to perform, and they're used to determine low molecular weight analytes in serum and urine, hormones, therapeutic drugs, and drugs of abuse. Rapid immunoassays are membrane-based, single-use, and disposable. Our pregnancy tests, some of our at-home COVID tests, these are those rapid immunoassays. They involve antigen or antibody being coupled to the membrane, are read by looking for a colored reaction, and may require the separate addition of patient sample, wash reagent, label antigen or antibody, and the substrate. So basically what happens is you're going to have a control line. The control line contains antibodies against human. So here's anti-IgE antibodies, and that's going to be there. And then you have the test line, which will capture the particular antigen or antibody that we're testing for. So once we add this to it, it's going to capillary flow down the test strip. We're going to add the clinical sample. The clinical sample is going to wash these antibodies. It's going to come down here, and then if the antigen is present in the clinical sample, it's going to bind to this antibody, and then the labeled antibody is going to bind, producing the color change. And then it's going to keep going, and this is going to bind to the sample. If the control line is colored, that means that the test is valid. If the test line is colored, that means that the person has what we were testing for. Fluorescent immunoassays, or immunofluorescent assay, is restricted to qualitative observations involving the use of fluorescence microscope. It involves grading the amount of fluorescence against a dark background. It's used for the rapid identification of microorganisms in cell culture or infected tissue, tumor-specific antigens on neoplastic tissue, and transplantation antigens. Direct immunofluorescence means that an antibody is conjugated with a fluorescent tag and it's added directly to the unknown sample. After incubation and wash step, the slide is read using a fluorescence microscope. This technique demonstrates the presence of pathogens in patient samples. Indirect immunofluorescent assay, patient serum is incubated with a known antigen attached to a solid phase. The slide is washed, and then anti-human immunoglobulin containing a fluorescent tag is added. A sandwich is formed with the first antibody, which localizes fluorescence, and the antibody can be identified. So here's direct. We have patient antigen on the slide, and then we directly add a fluorescently labeled antibody. We wash off any unbound antibody, so if the antigen is present, it'll stick and it won't wash away. If the antigen is not present, it'll wash away. If it's stuck, this fluorescently labeled antibody will start to fluoresce, and we can detect that in a fluorescent microscope. In this example here, we put known antigen on the slide, and then we're going to test the patient's serum for the presence of the antibodies. So we're going to see if this patient was exposed. So if the patient does have the antibody, it's going to bind to the known antigen, and then we have this secondary antibody that is labeled. The label will then fluoresce, and it's detected. If this antibody is not found, all of this would just be washed away and there would be nothing fluorescing. Fluorescence polarization immunoassays, FPIAs, used to determine the concentrations of therapeutic drugs and hormones based on the change in polarization of fluorescent light emitted from a labeled molecule when it's bound by antibody. If the labeled molecule is bound to antibody, the molecule emits an increased amount of polarized light. And this is a picture of that emitted light happening. Chemiluminescent immunoassays are employed to follow antigen-antibody combination. Involve the emission of light caused by a chemical reaction, typically an oxidation reaction, producing an excited molecule that decays back to its original ground state. It can be used for heterogeneous and homogeneous assays because labels can be attached to either antigen or antibody. And you can read through the summary slides.